This is Breakthroughs, a podcast from Northwestern University Feinberg School of Medicine. I'm Erin Spain, host of the show. This spring, the World Health Organization began using the Greek alphabet to label key variants of SARS-CoV-2. There are currently 4 variants of concern: Alpha, Beta, Gamma, and Delta, and 5 variants of interest: Eta, Iota, Kappa, Lambda, and Mu. The Greek names make it easier to talk about the variants with the public, but in the scientific community, these variants are being discussed and studied at the molecular level to learn as much as possible about their evolution, replication, and mutation. Here with insight on SARS-CoV-2 variants is Judd Hultquist, an assistant professor of medicine in the Division of Infectious Diseases at Feinberg and the associate director of our Institute for Global Health's new Center for Pathogen Genomics, Microbial Evolution. Dr. Hultquist is an HIV scientist whose lab has shifted many resources to study SARS-CoV-2 and track the origin of its variants, and also understand how antiviral proteins found in humans can protect against SARS-CoV-2 and other viruses. Thank you so much for being here.
Oh, thank you. I'm delighted to be here.
I'm wondering, what has it been like for you as an infectious disease scientist since the pandemic took hold in early 2020?
It's been a ride for all of us. And so it's been particularly interesting from my perspective, because I'm coming from it, both as a person living through the craziness of lockdowns and all of that's happening in social life, but also from a virologist perspective, it's truly been fascinating. Viruses jump into the human population pretty regularly. We've seen it with flu. We've seen it with Ebola recently, but SARS-CoV-2 coming in at this scale and having this much of an impact on global society, it's been scary and humbling, but at the same time, scientifically, absolutely amazing to be a virologist and witnessing it at this time.
You've been working consistently all the way through since the beginning of the pandemic. Tell me about that.
This was started all in February of 2020. And we had a meeting in the Division of Infectious Diseases that I'm affiliated with a few of the different PIs in the department. We were trying to decide, how should we prepare for SARS-CoV-2 if it turns up in the United States? We were still at that point. I know, right? Pretty naive. All in a room without any masks, just kind of casually discussing it. And so we asked permission from the hospital to gain access to some of their residual diagnostic samples so that if somebody did come in and they were tested positive, that we would get just sort of the leftover sample. And we would use that then to sequence the viral genome to see how the virus might be changing over time and how those changes could influence the clinical outcomes. And so that's really where we began. And little did we know how important viral genomes would become and how big this pandemic would be. And so it's been a whirlwind ever since.
It's pretty incredible foresight that your lab had, because at a certain point, you began tracking the epidemiological origins of the virus in our hospitals here at Northwestern and in Chicago as a whole. Tell me what happened once the virus did hit Chicago. Mm-hmm. What was your lab's role?
Yeah. So right when the virus began to spread in the Chicago area, you remember, of course, everybody kind of going into the lockdown phase. But we kept a skeleton crew here in the lab collecting these samples and running sequencing. And right from the get-go, from our earliest samples that we found in March, we found some really fascinating findings. For one thing, unlike the epidemic in so many parts of the globe, the epidemic here in Chicago was actually caused by 3 separate introductions of SARS-CoV-2, and all of them were different from one another. One of the viruses came from the East Coast, one of them came from the West Coast, and one of them came directly from China, we think courtesy of O'Hare Airport. And one of the cool things about that is by Chicago acting as this kind of melting pot of viral diversity, we could compare these viruses head to head within our own hospital system. And so we were able to look at the clinical data and the viral load data and all of the other data that is being collected by the hospitals on COVID patients. And what we saw was that depending on the strain that people were infected with, there were differences in how the virus behaved. And in particular, the strain that was coming out of New York was resulting in really high viral loads in people's upper airways, so in their nasal cavity. And what's important about that is how the virus is transmitted. And so that was one of the very earliest signs that the virus was changing to spread better in the human population.
And this work has really continued. And even now, all COVID-19 tests available to the public can detect all the different variants, but they can't tell you which variant you have. So in your lab, how do you decipher the variants in the patient samples that you obtain?
Yeah, very good question. So most of the diagnostic tests that people are using in the clinic are PCR based. And so that means that they're looking for a very specific part of the genome. But these viruses vary oftentimes at very specific parts of the genome that are not looked at by these tests. So what we do is we use a technique called viral whole genome sequencing. And essentially what we try to do is decode the entire genome for the virus for each patient that comes through the hospital. And to date, we've been able to do that for about 2,500 independent isolates. It's been a massive amount of work, but it's just an absolute goldmine of data. And based on what mutations are present in those isolates, we can assign it a designation. So alpha, delta, etc.
How have advancements in technology, computation, and modeling helped your lab and scientists around the world do the kind of work you're doing to study COVID-19?
Actually, that has been one of the biggest surprises, but also, biggest rewards out of this pandemic for us as scientists is all of this collaboration, not only between labs in different parts of the city, but across the state, internationally, labs that really specialize in different disciplines and different technologies. It has been such a concerted effort and has been so rewarding to be part of that because it's led to the development of all of these new types of technologies. And specifically, the sequencing work that we've been doing really got its start in some of the previous epidemics. So people have used whole genome sequencing to look at HIV and influenza in the past. And so we were basically able to adapt those platforms to now work with SARS-CoV-2.
And these tools will stay in your toolkit, hopefully after this pandemic is over, that those will be new tools in your toolkit you can use to continue investigating other diseases.
Oh, absolutely. And that's really what we're hoping to do. That's part of our vision in the Center for Pathogen Genomics and Microbial Evolution is to take the same approach and apply it more broadly to the pathogens that we're seeing in the clinic. So that can be anything from respiratory viruses to antimicrobial resistant bacteria. The same principles apply. You just need to adapt it, tweak it, and then really use it in these clinical settings to address diagnosis, treatment, and outcomes.
And I want to talk a little bit for some clarification about the variants of concern and the variants of interest as classified by the WHO. How are they different?
Yeah, so the difference is kind of subtle. So a variant of concern is a variant that is expanding in multiple parts of the globe and carries mutations that have been linked either to increased disease severity or increased immune evasion. So that means it has a mutation in it that has been proven to either result in less antibody efficacy, less treatment efficacy, or it causes somebody to get more sick when they're in the hospital. And a variant of interest, just in comparison, is a variant that has the potential to expand and has mutations that would be consistent with immune evasion or increased disease severity, but it hasn't been scientifically proven yet. So they're kind of the watch list for variants of concern.
Okay, so now do you use these Greek names in your research, or is there a different nomenclature system that the scientists are using?
Oftentimes, we actually do rely on those nomenclature because, as you know, it's changed multiple times. And it's been a problem because in sort of classical virology, people would name strains based on the city or country that it came from and the year. And the issue with that, and we saw a lot of this, especially early in the pandemic, is it's very easy to take that name, that location name, and imprint it with some kind of moral value about that's where that virus came from. Really, it could happen anywhere at any time. And so we really want to kind of remove that stigma of using location names. So then we came up with all of these other possible naming systems. You can name it based on the phylogenetics, which is basically the historical ancestry of the virus. And then it has these long letter number names like B117 and B1525. But those also can change over time. And those are quite a mouthful when you're trying to communicate it to the general public. But even if you're in Slack or on email and you're trying to explain to somebody, that's a lot of numbers and letters to be typing out. So we rely on alpha, beta, delta all the time.
So tell me, which variants and variant origins are you investigating right now in your lab, and what insights can you share with me?
Well, one of the most exciting efforts that we have ongoing right now is we're trying to do more sequencing for international surveillance. So while here in the US, we're very good about sequencing a representative sample of the isolates that come through our clinics, that's not true everywhere across the globe, either due to lack of resources or a lack of political will. And so we've been working with the Institute for Global Health to do more sequencing on an international scale. And in particular, we've been working with partners across equatorial Africa to do some sequencing in several countries there, including Mali, Guinea, Nigeria, and Tanzania. What we found in Nigeria specifically was while we were sequencing, we were getting more and more of this particular virus mutation set that people hadn't seen elsewhere in the world. And eventually it led to this new classification of this virus in West Africa as the Eta variant. We sequenced enough of them that the CDC had decided to classify it with its own designation. And what was insane about the Eta variant was that in Nigeria, it was competing head to head with Alpha, and it was outcompeting it. So it was even better than Alpha. But what we've been finding, of course, in our more recent sequencing is Delta came, wiped it all away. But that Eta variant of interest, it would have gone completely unnoticed if we weren't doing that kind of surveillance. And so unless we're paying attention to all of these undersampled regions across the globe, we can miss really critical variants that are arising in different places.
And what about the Lambda variant? You've also been doing some studies on this variant. What can you tell me?
In a separate project, we've been sequencing viruses from people who are here at Northwestern Memorial Hospital who have been in the intensive care unit for long periods of time. So these are people who are immunosuppressed, they can't clear the virus, and so the virus just continues to replicate.
Long COVID.
Now, long COVID, exactly. And what's interesting about long COVID is the longer the virus is in the body, the more chances it has to mutate and adapt to the immune response. And so we've been sequencing viruses from these patients periodically over time, sometimes over the course of several months. And we have 2 different patients now where we've been tracking the virus changing in them over time and seeing large deletions suddenly appear in their spike protein. Now, that's very bizarre because the spike protein is essential for the virus to replicate. The spike protein is that outer surface protein that attaches the virus to a cell. And so if you're deleting large portions of it, it's odd that that virus would really be functional at all. But by cloning out that virus, we were able to find that that deletion actually allows the virus to continue replicating, but makes it much more resistant to antibodies produced against that region. And the even more crazy thing is that that deletion is almost identical to the deletion that was found in the beta variant in South Africa. So what we actually think is happening is that some of these new variants of interest and variants of concern are originating from people with long COVID who give the virus the opportunity to evolve and adapt to the immune system. And then they escape outside that person and cause a larger epidemic.
So people with some of these folks with long COVID, they're going back out in the community and possibly infecting people. And maybe even more important for those folks to stay isolated and mask and do all those protective measures.
Exactly. It is vital that they adhere very strictly to, social distancing and masking guidelines. And beyond that, that they're getting tested regularly. If you're not producing virus actively, then you're not a threat. So if these people have been cleared and they're not actively producing virus, they're okay. And I want to be sure that we're distinguishing between people with long COVID and people who are long haulers. So people who have long COVID, they can still produce active virus. This is virus that's replicating within a person that could potentially infect other people. But there are other people known as long haulers who no longer are producing active virus, but they're experiencing symptoms in the long term. This is due to long term immune dysfunction. Those people are at no risk to anybody else because they're not producing any infectious virus.
So, do we know how long COVID typically lasts?
That's a great question, and I wish that I had an answer for you. The issue is that some patients just have struggled to clear the virus. Sometimes it will go away and the person is absolutely fine. And sometimes patients end up succumbing from long COVID. And at this point, it's hard to guess, but that's one of the reasons why we're continuing to research it, because we're hoping that then we can gain insights into why do some of these people stay sick for 2 months and then get better, and some of them don't.
So there's people out there, like you said, with the long COVID who stay sick for a really long time. And then there's some people that seem to somehow be protected against SARS-CoV-2. And in your lab, you're doing research on antiviral proteins in humans that can protect against SARS-CoV-2 and also offer pathways for potential therapeutics. Tell me about that and what you've discovered.
Yeah, it's something that we really don't think about all that often. I guess people used to go into the office, and a bug would be going around and Susie next door might get a little bit of a head cold and you're just fine. But then Tom in the cubicle over is out for 2 weeks. That kind of variation in how we respond to pathogens is remarkable. And we know very little about it. We do know that it can be influenced by past exposure to a pathogen, but that certainly can't explain SARS-CoV-2, especially at the beginning of the epidemic, because it was completely new. So part of the 10,000-foot view questions that my lab really is interested in is asking is how differences in hosts, that is differences human to human, can drive differences in disease severity and pathogen replication. And that's really important because when you think about non-communicable diseases, so these are things like cancer, neurodegenerative disorders, autoimmunity, it's all about human genetic diversity. But when we think about infectious diseases, we know very little about how human genetic diversity influences replication, outcome, treatment. And that's one of the links that we're trying to make. So what we found with SARS-CoV-2 is that there are a large number of proteins in the human body that actually act as part of the immune system to try to interfere with the replication life cycle. And what we're hoping is that we can identify changes in those genes that make people either more or less susceptible to infection.
So before the pandemic, many of your publications were about HIV. Have you been able to continue this important work?
Yeah, and that's a wonderful question because I really got into the whole field of infectious diseases because of HIV. It's a fascinating virus, and that's really what I thought I would be dedicating my career to. But then, of course, SARS-CoV-2 comes along and the world is suddenly needs a lot of virologists. And so a lot of my colleagues in HIV all jumped and joined the SARS-CoV-2 effort. So right now, we're actually back to about 50-50. About half of my lab is currently doing SARS-CoV-2 work, and half of them are doing HIV work.
Tell me about the parallels between HIV and SARS-CoV-2.
People have looked back to the HIV pandemic as a way to understand what has been happening with the SARS-CoV-2 epidemic. So when we think about where HIV came from, for example, it was due to zoonotic transmission from primates into humans. And now when we're trying to understand where did SARS-CoV-2 come from, of course, the first answer to that question would be it's likely a zoonotic transmission. And unlike primates, where we can find these long-lasting lentiviruses, here we're trying to catch these really short-term evasive infections in species of bats. Unlike primates, are flying around and there are millions and millions of them. So trying to find the exact precursor to SARS-CoV-2 has been a challenge. But that's just regarding the origins. Everything from how do we diagnose SARS-CoV-2 was informed by early lessons we learned in the HIV epidemic. How do we apply public health measures, social distancing? The same principles applied there. And even how we're researching it was informed by a lot of what we've been doing in HIV. In fact, a lot of the centers and institutes that were originally designed to fund and support HIV research have been converted or have, played essential roles in supporting SARS-CoV-2 research. So it really has been driven by scientists in these communities who decided, this is a major challenge. We got to come together and tackle it. And I've been so so grateful and amazed at all of the people who've been willing to do all of that work.
Yeah, tell me, here at Northwestern, New Cats, the Institute, has been very supportive of your recent work. Tell me about that.
Yeah, New Cats has been absolutely phenomenal. They originally came out and said, hey, we want to fund people to start working on SARS-CoV-2. They wanted to drive innovation. They wanted to drive collaboration. And we actually received one of their earliest pilot grants that actually kickstarted our biobanking effort. In the grant that we wrote for New Cats, we said we wanted to collect 300 SARS-CoV-2 specimens and sequence almost 100 of them. And that was our big vision. And we're so thankful that they did, because now we're up to over 10,000 specimens banked and over 2,500 sequenced. And it all really came down to the funding from New Cats at the beginning.
Annually, about 15% of all deaths worldwide are directly attributable to infectious diseases. And this is according to the WHO. Do you expect that number to increase or decrease in years to come?
That's another excellent question. I would have to guess that infectious disease deaths are going to decrease going forward. They've been decreasing pretty steadily up until the outbreak of the COVID-19 pandemic. But most of these outbreaks are preventable. They're due to diseases that we have vaccines against, or we have public health measures that we know can protect people. It's about getting the resources to where they're needed. And there are so many amazing global health efforts that are ongoing today to try to address these challenges, from tuberculosis to malaria to HIV and SARS-CoV-2. And so while new epidemics and pandemics are constantly going to be arising, especially as climate change, globalization, urbanization continue to influence the number of species and viruses that we interact with, I still think on the long scope of things, we're heading in the right direction.
Great note to leave us on today. Thank you so much, Dr. Judd Hultquist, for joining me today, sharing some information about what's been happening in your lab over the past over a year and a half now. Thank you so much.
Absolutely. Thank you so much for having me.
Thanks for listening, and be sure to subscribe to this show on Apple Podcasts or wherever you listen to podcasts, and rate and review us. Also, for medical professionals, this episode of Breakthroughs is available for CME credit. Go to our website, feinberg.northwestern.edu, and search CME.